Bioinfocongress IV Associate Professor Dr. Pınar Uysal Onganer, microRNAs as Biomarkers in Cancer, Opportunities and Challenges. Associate Professor Dr. Pınar Uysal Onganer is a reader in molecular biology and cancer mechanism research group leader at University of Westminster, London, UK. She has finished her PhD by completing her studies both at Center for Hepatology at UCL and the Medical School of Marmara University in Turkey. Her research experience involves a range of cell signaling mechanisms such as VNT signaling, microRNAs, and the functional and molecular aspects of cancer. She has also been actively involved in fundraising activities with charities such as the Cancer Research UK, the Pancreatic Cancer UK, and the Prostate Cancer UK. She is an Ethnaswan coordinator of University of Westminster and runs team activities for local schools, and elected as Fellow of American Chemical Society and an ambassador of EACR. Dr. Uysal Onganer completed Prevention of Cancer and Control Summer Curriculum at National Cancer Institute, NIH, at 2017. She became a Fellow of HEA at 2014 and Senior Fellow at 2018.
Dear Associated Professor Dr. Pınar Uysal Onganer and participants, welcome all of you to our Bioinfocongress IV event. Associate Professor Dr. Pınar Uysal Onganer will give us a talk today on microRNAs as biomarkers in cancer, opportunities and challenges. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Associated Professor Dr. Pınar Uysal Onganer, welcome back.
Thank you.
Thank you so much. If you wish, you can start your presentation.
Trying. Yes, here we go. Thank you so much for this invitation, and good afternoon, everyone. Thank you so much for being here for this. I don't know how is the weather where you are, but I guess it's a very nice Sunday afternoon. So I am going to talk about microRNAs today, and I'm going to give a couple of examples of my lab, what we are doing, why we are doing those experiments, those projects. So let's start. So sorry to start with slightly, I guess, disheartening slide. According to the CR UK, let me just make my slide bigger. According to the CR UK, 1 in 2 people in the UK, this is not only about the UK, obviously, but like in general and worldwide, will get cancer in their lifetime. In an extremely basic explanation, as I'm sure you all know that the cancer is abnormal cell dividing, and some cancer may eventually spread other tissues. So the good news is we do have lots of very clever techniques over the years that we came across with lots of different challenges when it comes to cancer. However, we developed a lot brilliant opportunities to tackle cancer. Once again, very briefly, if there's a cancer, if there is a solid tumor, especially I'm talking about, we can easily cut the tumor, that is surgery. And then we can poison the tumor, that is chemotherapy. And then we can burn the tumor, that is radiotherapy. However, the cancer is still a huge global health problem, because most of the times it's not the cancer itself, but the metastasis kills people. Let's look at what metastasis is. In this, once again, extremely basic slide, you do see the primary tumor formation. In here, there are very abnormal cell growth we can see. However, they are still like very compact. And then when we move on to the local tissue invasion, obviously after a while, these cells, because they just keep dividing abnormally, there is not enough nutrients and there is not enough space. Therefore, they need to jump on this is a local tissue invasion. And then early into the vessels, they do need to jump on a vehicle, in other words. And this is most of the times a bloodstream. And then they do need to survive in the bloodstream, and then they need to move on to the different parts of the body. And this is what we call metastasis. Once they are escaping from the bloodstream, and then they start new colonies in the different parts of the body. So this is, in fact, is the problem when it comes to cancer treatment, because, let me just go back, because as I tried to very basically explain to you earlier on, if we know where the cancer is, once again, I'm talking about the solid tumors, we can easily tackle that. We can just remove the tumor, and then we can just use either chemotherapy or radiotherapy, and then we can just see whether the patient is going on healthily. However, most of the times, this escaping cells, tumor cells, jumping on the vehicle, the vessel's bloodstream, and then moving on to the different part of the body, it takes time. And most of the times when we diagnose people with metastasis, it is already too late. So it is already spread all the other parts of the body. So this is the actual issue with the cancer itself. So my lab focuses on cancer metastasis. We are basically trying to understand the mechanisms of the cancer metastasis. Moreover, we are trying to find out better biomarkers to understand which tumors are going to metastasize. And if we can come up with better biomarkers, of course, we're hoping that this will also open more doors when it comes to the therapeutic approaches. So one of those things that we have been focusing on, one of those potential candidates, is microRNAs. Well, microRNAs, hence the name, they are non-coding small RNA strands, single strand RNA molecules. They are around 18 to 20 nucleotides long, especially since the Human Genome Project, as again, you know, it is completed around 2003. And we found that completion of the Human Genome Project, huge part of our body is not required for protein synthesis. So since then, we are trying to understand why do we have this, we used to call them junk, we don't call them junk anymore, this big part of our genome is what is happening? Why do we have this non-coding part of our genome? So one of those parts is microRNAs. The other one is, these are non-coding RNAs, the other ones are link RNAs, long non-coding RNAs. Today, I am going to talk mostly about the microRNAs. As I said, under the normal conditions, these microRNAs are responsible for controlling the cell growth, proliferation, differentiation, and the cell death. So they are it's been shown that one microRNA, as I told you, they are quite small, single-stranded RNA molecules. They can control almost 100 different mRNAs expressions. You can see that they are quite small, but they are quite powerful. So if something goes wrong with the microRNAs, this time we do have problems such as cancer. So we know, again, because of the research, we know that some microRNAs, they behave like tumor suppressors. We call them tumor suppressor microRNAs. And some of them, they do behave like oncogenes. That's why we do call them oncomers. So as a summary, like a long introduction summary, one microRNA can moderate the gene expression at the post-transcriptional level. They can bind to the complementary sequences in their target mRNAs, especially from the 3' untranslated regions. Due to the interactions between the 3' end of the mRNA to microRNAs, they can control the expression of several mRNAs. So that means they can also involve in several signaling pathways. And also, it's been found that they do have significant role in several biological processes. Those processes, as I tried to show you in the previous slide, cell differentiation, cell survival, proliferation, invasion, apoptosis, angiogenesis, metastasis, and lipid glucose metabolic pathways. So only this part, if you see, we are already talking about cancer. If something goes wrong, so all of these things are going to affect it. So therefore, the cells, the cancer cells, are going to behave more aggressively. So let's start looking into the projects that we have been doing over the, I guess, like 5 to 6 years. So the first project is breast cancer and microRNAs. In this introductory slide again, I'm just trying to show you where the stroma, where the duct is. So you know, again, I'm sure the breast cancer is the second biggest killer in the world, and it is the biggest killer amongst women. We did have a lot of success in terms of the breast cancer treatment over the years. There are most of the countries, they do have screening programs. However, the breast cancer is quite, as all the cancers are, quite heterogeneous. So we do have hormone dependency and we do have hormone independency when it comes to the breast cancer. Obviously, these hormones are progesterone, estrogen, and EGF hormones. So what I'm trying to show you here, if you look at the normal breast, you can see the stroma, you can see the duct, and I hope you can appreciate there is a beautiful differentiation, and it is beautiful, in a way, order between the stroma and the duct. When we look at the benign lesion, still there is an abnormality happening. You can see that the hyperplasia region in here, you can see that there are lots of cell division occurs, but you can still see this like beautiful ordering. So the differentiation, in other words, maintained in here. Whereas when we look at the malignant breast cancer picture on my right-hand side, you can see that it's just like in a way it's a mess. So you can see all those like green bits, they were like stroma, remember? And then the duct, so they sort of like divided and they sort of like in between each other. So this is the actual problem that we are trying to understand in my research. So in this particular project, we just wanted to see microRNA 21, which occurs or which behaves like an oncomer. So in other words, we do see the higher expression of microRNA 21 in pretty much all of the cancers. We wanted to see whether the microRNA 21 is overexpressed in the breast cancer. If so, does it really do anything in terms of the overall survival? For this purpose, we went to the TCGA breast cancer database and we wanted to see whether there is any correlation between the MER21 expression and the overall survival in almost like 1,000 patients in total, 995 to be exact. So in the left-hand side, you can see the Kaplan-Meier curve. And what you should look at, so in the red bits, you can see that if there is a higher microRNA expression in the patients, they don't survive as much as the patients that they do not have higher microRNA 21 expression. So this was our starting point. So then we wanted to go back to the lab. So we wanted to see whether we are going to see any hormone dependency. So by using the cell lines, MCF7s, SKBR3, and MDA231 cells, these cell lines are very, I guess, like popularly used in all around the world. So we did see the triple negative breast cancer cell has the highest microRNA 21 expression. This pretty much fits our hypothesis because this microRNA 21 higher expression we did see in MDA-MB231 cells. This cell line is, as I said, triple negative, but most importantly, this cell line has the most metastatic ability when you compare to the other breast cancer cells. For instance, this MCF7 cells, they are also metastatic, however, they are very weakly metastatic because they also have the hormone dependency. So we did use MDA cell lines as a cell model to be able to understand the role of microRNA 21 in breast cancer. So by using the CRISPR gene editing technique, we targeted microRNA 21, and then you can see in this bar chart next to it. So when there is a GFP or the control control MDA-MB23 cell lines, and then we have like 4 different guide RNAs. We transfected the same cell line with 4 different guide RNAs, and then we found that 2 of our clones they show the lowest microRNA 21 expressions. So from now on, we use the clone 2 and clone 4. So another important area when it comes to all, the cancer metastasis, and also the cell signaling, as again, I'm sure, none of the cells, they can live in isolation. They do need their neighboring cells to be able to communicate with the neighboring cells. They do need to express, they do need to secrete certain factors. So therefore, the secretion and then endocytosis are quite important in terms of the healthy communications between the cells. When it comes to the cancer, this becomes even more important because we know that the one cancer cells, they can secrete growth factors, the proteins that the other cell lines, the neighboring cell lines, can also take of these ones, and then they can behave almost like the cancer cells as well. Just to test this hypothesis, we wanted to look at the extracellular vesicle release and the cargo change, whether the cargo is changed when we CRISPRed, when we remove the microRNA from the MDA-MB23 cells. For this purpose, again, we looked at both of the clones 2 and 4, whereas in here you can only see the example of the clone 2 versus control. What do we see from this image or this slide? I hope you can see it in here in the D. Total EV release reduced when there is no microRNA 21. So with the control cells, they did have like more release, extracellular vesicle release, whereas if there is no microRNA 1, the cells release less extracellular vesicles. Moreover, the cargo, when we extracted these vesicles and then looked at their cargo, what they carry, when we found that the microRNA 21 CRISPRed cells had a lot less microRNA 21 than the controls. So so far it was good, but we wanted to see what does it mean. So is this going to affect the cell growth, for instance? So we looked at the cells again. So once again, you do see in here the wild type and one of the examples of the knockout cell lines. We just started to grow the cells, but we sort of looked at them in 48 hours to 72 hours, in 2 different time points we use. If you look at the top panel in here, you can see that with the wild type MDA-MB23 cells, you can see that the cells are growing like colonies, and you can see that they are sort of started to spread around. Whereas with the no microRNA clones, CRISPRed clones, you can see that they sort of stuck in compact. So they are still growing, but they cannot spread as much as the control. As I said, this experiment only done up to 72 hours. We didn't move more than 72 hours. But even in 48 to 72 hours, we started to see the difference between the wild type versus the knockout clones. Another marker that we use when we look at the metastasis, EMT phenomena. So EMT stands for epithelial mesenchymal transition. It is important because, again, mostly in solid tumors, in the epithelial tumors, we do see the epithelial cells are really nicely elongated, and they are, as I said, they do need their neighboring cells. They do have connections with the neighboring cells and also the surface. So if the cells are looking and behaving like epithelial cells, the marker for them is E-cadherin. If during the cancer, we do see this. If these beautiful looking, well-behaved epithelial cells going through the dark side, this time they do have like less E-cadherin expression, but they do have more mesenchymal markers expressions. Those mesenchymal markers are, in here we use snail, zeb, and vimentin. But mostly in the literature, people are using E-cadherin and vimentin as a marker of the EMT epithelial mesenchymal transition. In this slide, hopefully you do see with the control cells, there are almost no E-cadherin expression, meaning that these cells are already quite aggressive. They don't need any connections between the neighboring cells, therefore the E-cadherin expression went down. Whereas when we remove the microRNA from these highly metastatic triple negative breast cancer cells, we did see that the E-cadherin expression started to occur, hence the greenish signaling here. So when we move on to the mesenchymal markers, as I said, we use snail, Z1, and vimentin. Let's look at the vimentin. With the MDA-MB23 cell, we did detect high level of mesenchymal marker vimentin. This was similar to the snail and Z1. However, when there is no microRNA 21, this mesenchymal markers started to disappear. So the expression level of the snail, ZEB1, and vimentin reduced. So this was quite an important result for us. So we moved on to the invasion markers. We showed that when there is no microRNA 21 invasion, matrigel invasion also reduced. So we published this data, I think it's almost 2 years now. And currently we are trying to generate another cell line and trying to understand whether this microRNA 21 is working in the similar line with another triple negative breast cancer cells. So this was the breast cancer. Let's look at the prostate cancer, whether we are going to see the similar approach on prostate cancer. Let's start with the stats again. According to the Prostate Cancer Research UK, 1 in 8 men over the age of 50 is diagnosed with prostate cancer. Prostate cancer is similar to the breast cancer in terms of the hormone dependency. This time our hormone is androgen, not estrogen, but androgen. But there is also something different from the prostate cancer. Most of the times, or at least until very recently, the approach was watchful waiting. So in other words, the clinicians were saying that, well, the prostate cancer is, you can see it in here, so mostly is diagnosed in the men over 50 years old, and it is a mostly slow growing cancer type. So instead of like interfering, instead of using our clever techniques that I mentioned to you earlier on, let's just like keep observing whether the cancer is metastasizing or just like being more aggressive. So this was the approach for a while. However, they found out very recently this is not working really well because more and more people are now dying from prostate cancer. The reason for that, the main reason for that, we still don't have a really good biomarker for prostate cancer. There are like 2 ways to diagnose prostate cancer. One is the digital rectal examination. Another one is the blood test called PSA, prostate specific antigen. Everyone knows that the PSA is not specific enough. It doesn't show us, it doesn't give us any more information than the high or low. However, this is still the case in, again, all around the world when it comes to the prostate cancer detection. So when we look at the molecular level, so you can see that the progression pathway for human prostate cancer. So this time, again, very similar, I believe, for the previous example. You can see that in the normal epithelium, these cells are like beautifully aligned in here. And then we move on to the metastasis. We do have like different stages. The first one is the PIN, prostatic intraepithelial neoplasia. You can see that between the normal to the PIN, there are like certain genes activated. So most of one of them is the very important one is the P10. P10 is a tumor suppressor. Almost all of the prostate cancers, we do see the P10 inactivation. Another important one is TMPRSS-ERK fusion. So two of the genes, TMPRSS and ERK, they do break and they become a fusion. And again, like almost 60% of the prostate cancer patients, we do see the TMPRSS-ERK fusion. And then we have the adenocarcinoma. I'm sure you already noticed that the cells are becoming like a bit more aggressive, and they started to go out from the normal epithelium, and then adenocarcinoma, and you can see the metastasis. And most of the time when the metastasis occurs, the treatment doesn't work. So we start to see the castration resistance, we started to see the EMT markers again, E-codering goes down, mesenchymal markers such as vimentin goes up. So this is what happens normally. So we wanted to see if the P10 deletion in the prostate epithelium is correlated with the microRNAs, or whether the microRNAs are doing something with the P10. For this purpose, with the collaboration of the Institute of Cancer Research here in the UK, we use P10 knockout mouse model. This is not the human study, this is the mouse model study. So for this, for the mouse, if there is no P10, they don't completely grow for the metastasis or the prostate cancer, but they sort of like stuck at the PIN interplasia, which was a lot better for us because we wanted to see, especially with the P10 involvement, whether the microRNAs are also the early event of the prostate interplasia. So for this purpose, again, when we have this mouse models, we just took their tissues and then we generated the sections of the prostate tissues. And you can see that this is a very long, big paper, but I just wanted to show you this. For this paper, we looked at the wild type versus the P10 knockout mouse models. We wanted to look at the microRNA 21 and microRNA 155, which is another oncomer, behaves like oncogenes. So we did see that with the wild type, almost like there is no change in terms of the microRNA 21 and 155. Whereas if there is no P10, both of the microRNAs are highly upregulated. So our aim was just to look at the inflammatory pathway as well as a response to the carcinogenesis, because in cancer, most of the times inflammation goes hand in hand with metastasis or cancer. However, we didn't see this, but the important part of this paper, we found that there is oncomers such as 155 and 21 show a significant effect of the gene expression profile. When we inhibit the P10 signaling cascade, we did see the reduced expression of the microRNAs significantly. And we've also shown that the microRNA inhibitors to reduce the cell growth and to re-express several and the previously downregulated genes. So there is a clear association between these two microRNAs, microRNA 21 and 155, and the P10 expression in prostate cancer. While this study was going on, I was also trying to optimize a method in the lab, which was the microRNA in situ hybridization. So for this purpose, we already had human cells and human tissues, prostate cancer tissues. So if we first wanted to be able to optimize it, we first wanted to look at the Gleason 7s. I didn't mention about the Gleason. So the Gleason scoring is, again, another routine technique in the histopathology labs. So if mostly we do need to see if the Gleason 7, if the patient diagnosed with Gleason 7, you can see that there is a prostate cancer, but this is mostly localized. So the cancer is there, but it is localized in the prostate, and it is no metastasis so far. So if there's a Gleason 7, like lower than Gleason 7, this is in a way better, because this time it is a very early diagnosis of prostate cancer this patient has. If there is a bigger than Gleason 7, that means that cancer is already advanced and most probably the hormone independent at that stage, and most probably it has already metastasized. So this is the how the clinicians use the Gleason 7 Gleason scoring. We first wanted to start with the Gleason 7, that is almost like the mid-range localized tumors. For this reason, we collected 20 patient samples. These are tissue samples taken with the needle biopsies. They were like used with the sectioning. And you can see that we did see in here as well, this is sort of like the purplish color that we do see, I hope. So with this purplish part, which is a prostate, and you can see prostate cancer, you can see that there is a higher microRNA 21 expression in the human patient samples. When there is a lower Gleason, when the prostate cancer is detected in an earlier stage, this time almost no microRNA 21 expression we did see. Whereas if you move on to the higher Gleason score, we did see a higher microRNA 21 expression. However, if you look at this chart, you can see that although the sample sizes are slightly different, so you can see that there's almost like a bell curve going on. So it's not like a straightforward curve. So you can see that there is a lower microRNA 21 expression with the lower Gleason score, which you would expect. And then when there is a Gleason 7, almost like a localized prostate cancer, you do see a higher expression. Whereas when there is more metastatic patient samples, you can't see even like higher. It's just like sort of drops. That sort of tells us perhaps the microRNA 21 expression triggers the prostate cancer, even the metastasis. But once the tumor reaches to the aggressive stage, it is not needed anymore because the tumor is already metastasized. So we also know that microRNAs can be released into the bloodstream by the tumor cells. So the apoptotic cells and the active cells, I've given you the example in the breast cancer. I told you that the cells secrete extracellular vesicles we looked at for the breast cancer project. So this is pretty much like the everyday event. So we do have so many secretions going on. And I also told you that with the prostate cancer, we have a problem in terms of finding a better biomarker. So for this reason, we just wanted to look whether the microRNAs can be detected from the bloodstream. These are the circulating microRNAs this time. So we have already shown that we can detect and we can show microRNA expression in the tissue samples, both in the mouse models and the human samples, and also obviously the cells. But we wanted to see whether we can just come up with potential biomarkers, better biomarkers, by using the non-invasive techniques such as like taking a blood. Again, it's not a brilliant image for the Sunday afternoon, I believe, but you also know that obesity and the high-fat diet promote a prostate cancer progression. So that means we have two aims for now, right? So one, we are trying to find a better biomarker, non-invasive better biomarker for prostate cancer, but also we wanted to see whether there is any association that we can detect in terms of the different microRNAs. Is there any microRNA that we can find out which is the obesity related? And then we already have a couple of markers, couple of candidates for prostate cancer. Can we make these biomarkers, microRNAs, to detect the obesity and the high Gleason score, obesity and low Gleason score? Right. This is a very beautiful experiment, I like to say. So we found that 69 volunteer patients for this project. So we wanted to look at, and they were like grouped in different parts. So for instance, 41 patients, they do have the high BMI. BMI is an index that body mass index that we do use to, again, decide whether someone is obese or not. There are lots of controversy in the clinic, in the scientific world. Most of the people are saying that the BMI is not a good marker for the obesity, because many of us, we do look like slim, but it doesn't mean that means we have a low BMI, but it doesn't mean that we are healthy, because there's a new term called skinny fat. So perhaps, in the outside, we look like quite slim. However, our internal organs like liver, they do have the high fat. So therefore, the BMI is not like really good way to look at whether someone is healthy or not. But forget this. For this purpose, for this project, we just wanted to use the traditional method. So for this one, 41 patients with the BMI lower than 30, and then we have like 28 patients with the higher BMI, and then also they did have the different Gleason scoring. So we looked at 5 different biomarkers, 5 different microRNAs. These microRNAs were pre-selected by doing lots and lots of reading, and also doing lots of database searches. So you can see that the high BMI and the high Gleason score, they did have like different microRNA expressions. So we also wanted to check whether the ROC curves are going to show whether these microRNAs are sensitive and specific enough. And this paper was already published, I think, almost like a year ago again. So this was another way that show the microRNAs can be used non-invasively as a biomarker, and distinguish between the not only the higher and the lower Gleason when it comes to the prostate cancer, but we can only distinguish by using microRNAs high BMI to the low BMI patients. So I think I have a bit more time, so I like to talk about the pancreatic ductal adenocarcinoma. This is the final project that I'm going to mention today. I'm going to sound horrible, but I told you that my lab is mostly interested in metastasis. So therefore, the most metastatic cancers are the better models for us. And pancreatic ductal adenocarcinoma is one of those. There are still like 4% overall survival rate for 5 to 10 years for pancreatic ductal adenocarcinoma. It is extremely aggressive cancer type. There is no biomarker, better biomarker. Most of the times when the patient's diagnosed with the pancreatic cancer, pancreatic ductal adenocarcinoma, the cancer already metastasized, and most of them, they don't even go through the treatments because it's already too late. So we first wanted to look at whether the certain microRNAs are involved in pancreatic ductal adenocarcinoma progression. So for this purpose, this is a review that we published a couple of years ago. So you can see that we sort of mapped all the known microRNAs in terms of the cancer hallmarks. So you can see that there are microRNAs that are associated with avoiding the immune destructions. There are certain microRNAs that involved in or associated with genome instability and mutations. Some of them are associated with inducing the angiogenesis. What I hope you see in here, there are always similar ones, the usual candidates, and the usual ones like in 155, which we talked about it, 21, which again we talked about it. It just keeps occurring in everywhere. So it seems like these 2 microRNAs, or like a couple of them in 210, which I'm also going to mention in a minute. So these microRNAs are like pretty much coming out in every single hallmarks in here. So we wanted to see first whether we can just shortlist certain microRNAs. So for this purpose, we used this paper. We found that there were like small number of patients, but this small number of patients, they did have both the tumor tissue, pancreatic ductal adenocarcinoma tumor tissue, and also the normal part of their pancreas. So they're, in other words, they are sort of like matched. They both are coming from the same patient. We wanted to see whether there is any signature comes out in terms of the microRNA expressions. So this is completely in silico study. We looked at these patients' RNA sequencing data, and we found out microRNA 21, 221, 375, and the other microRNAs, they sort of like highlighted in terms of the different patient samples. So we mapped out all the 20 tumors, so there were like 10 males and 10 females in this study, and also the downregulated microRNAs, and we created these charts as well. So you can see that there are certain microRNAs. Again, I'm not sure whether you can see it really clearly here, but the same microRNAs that we have been working already for other projects came out. So then we moved back to the lab. So we wanted to see what is happening with the in vitro cell line studies. So the HPD here is the normal epithelial human cells, and there are like 3 different pancreatic ductal adenocarcinoma cell lines. This time, unlikely of the previous 2 studies, there is no hormone dependency we're talking about. When you look at the literature, all the pancreatic ductal adenocarcinoma cell lines are extremely metastatic. However, the BAX-PC3, we found out they are like more epithelial than the PANC1. In other words, BAX-PC3 is less metastatic than the PANC1. So we looked at 5 different microRNAs for this study. So what you can see, and then we normalize all of these expressions in terms of the normal human duct epithelial cells, HPDE. And you can see that microRNA 21, which we have been talking since the beginning, is beautifully correlated with the metastasis again. 155, again, we've talked. Again, you can see that the PANC1 has the highest expression. 221, it is another important microRNA, another oncomer. You can see that it is, again, the similar thing we are seeing. There's an upregulation in the PANC1 when you compare to the HDPE. But when it comes to the 2 of the tumor suppressor microRNAs, the scene is completely reversed. So you can see that the PANC1 or the MAIA-PACA2, they do have the least expressions of 2 of the tumor suppressor microRNAs when it's compared to the normal duct epithelial cells. Okay, so we first started with the in silico analysis by using the RNA sequencing of the patient samples, matched patient samples. Then we moved on to the cell lines, and we wanted to see what is happening with these cell lines. Using the similar approach that we used before, this time, again, we looked at the 2 different cell lines, and we CRISPRed 2 different cell lines. And then you can see that when there is no microRNA 21 in both of the cell lines, microRNA 21 reduced. More importantly, all the cancer stem cell markers are also reduced. So the cancer stem cells are important because we believe that according to the seed and soil model, we believe that according to the literature, there are some cells that they are already in a dormant phase, and these mostly are the cancer stem cells. All of our brilliant, beautiful approaches like cut, burn, and poison is not working because they don't care the hormone independency. They are not hormone independent anyway. So I think I'm sort of like running a bit out of time, so let me just like speed up a little bit. So you can see that the microRNA 21 knockout diminished expression of all the cancer stem cell markers in pancreatic ductal adenocarcinoma. So like 5 different cancer stem cell markers we looked at in both of the MAIA-PACA and the PANC1 cells. And this was similar in the flow cytometry analysis as well. And then the invasion also reduced when there is no microRNA 21. So as you can see, the microRNA 21 is highly important when it comes to the cancer metastasis, not only for breast cancer, but also another highly metastatic cancer type in pancreatic ductal adenocarcinoma. So of course, we looked at more of the different signaling pathways as well, and then we published this paper. So if you like to read about it, it's already available. So another side project that I wanted to mention to you. So my lab, we are already interested in the in vitro studies and trying to understand the mechanisms of the metastasis. But we know that this is not only about the tumor microenvironment. There are like more important things, like because we all like live in an environment. So more and more we are started to, I guess, like interested in the exposomes. What is the exposome? So the exposome, by definition, refers to the totality of the exposures from the variety of the external and internal sources. So we have been already working on the internal resources, but we didn't look particularly the external resources as well, or the sources as well. So this is a word that I quite like. There is a quote in here: all illnesses have some sort of hereditary contribution. It's been said that genetics load the gun and the environment pulls the trigger. So our question was, what is happening with the environment? Or does the environment is affecting the microRNA expressions in our body? So for this reason, I'm not a toxicologist, but I have great collaborators. They are toxicologists. So we looked at whether we can use, the usual suspects, the microRNA 21, 155, both oncomers, and 126, that is a tumor suppressors, as a response to the environmental heavy metals, environmental toxins. So we published 2 studies on this. So we basically treated the cells in extremely low conditions, not toxic at all, cadmium and nickel. Both cadmium and nickel, we do see it, or we do expose a lot in our usual lifetime. Now, when we were writing this paper, for instance, I was quite scared because, for instance, the cadmium, we have a lot as a health risk in rice. So we do have lots of rice most of the time. Well, there are lots of cadmium in it. All the pulses, all the nuts that we use, especially if you're a vegetarian, to be able to get the protein source, you are taking lots of lentils, you are taking lots of the nuts, lots of cadmium as well. So again, I found it quite interesting to see that when we treat the pancreatic cancer cells with extremely low amount of cadmium and nickel, up to 3 weeks, we did see their expression levels completely went up. So perhaps we can also use the microRNAs as biomarkers for the environmental toxins. Right, the conclusions. Alterations in the microRNAs are involved in pathogenesis in various of the human cancers. In here, I talked about pancreatic ductal adenocarcinoma, prostate, and the breast cancers. I believe they have a great potential in terms of using them as a biomarker because they are quite small. They're only like 18 to 22 nucleotide long single-strand RNA molecules. They are highly stable. And we can use them from the bloodstream. So we can just look at the circulating biomarkers. So they have a huge potential to be used as a non-invasive diagnostic tool. I haven't talked anything about the therapeutic approaches in here, but they also can be used as a therapy as well. Of course, we do need lots of specific studies to be able to understand whether they are specific and sensitive enough to be able to use as a biomarkers. But there is, these are the opportunities, the challenges, I must say. The problem with the microRNAs, I'm sure you've noticed. So microRNA 21, we've seen the upregulation in 3 different cancer types so far. So it is not cancer specific. That is one of the issues that we need to consider. Another one is when we use the circulating microRNAs, we do not know where the primary tumor is. There is no particular specificity of this microRNAs, whether it is coming from the prostate, whether it is coming from the breast cancer. So we cannot associate, or we cannot differentiate this certain microRNA in terms of the certain site, so because we are using this as a non-invasive tool. So we started with a bit of a depressing slide by saying that one of 2 people are going to get cancer in their lifetime. But the good news is there is a high survival rate of the cancer right now, and I'm hoping that like in 5 to 10 years we are going to have even more survival rate. So we are going to have a lot more success than before. So as I said, I did not have time to talk about therapeutics, but already lots of labs are working microRNAs as a therapeutic targets as well. In this another review, we sort of mapped which microRNAs can be used as a therapeutic. In fact, we do know that there are certain clinical trials going on, and this is one of the examples. Miragen is one of the companies at the US. So they are looking at the microRNA 155 because similar to 21 story. So 155 expression is associated with the poor clinical outcomes. So therefore, they are trying to use this anti-MIR 155 drug for the blood cancers and lymphoma. So this is a FDA-approved clinical trial going on. And if you look at the FDA's website or any recent review, you can see that there are lots of clinical trials going on to use microRNAs as a therapeutics as well. And mostly the Miragen is doing this. Right, I think I have only 10 minutes left. These are the wonderful people that I have been collaborating recently, and the data that I showed to you was due to the collaboration between different labs and the different groups. Thank you so much for the invitation, and thank you so much to all of you to listen to me to talk about microRNAs today. Right, I'm going to stop sharing and I'm ready to hear if you have any questions. Thank you.
Thank you so much, Dr. Onganer. We would like to thank you for this informative presentation. I'm directing the first question. In treating the malignant disorder and preventing tumor recurrence, diagnostic and therapeutic techniques targeting these cells and their regulators, in addition to differentiated tumor cells, are required. Could sample size limit this study when working with microRNAs at this stage? Would it be advantageous to take a larger sample size from different populations?
Okay, if I understood correctly, the sample size. So the sample size definitely is so important because the main issue is cancer is like quite heterogeneous. In other words, that everybody's cancer is unique to that person. So we are trying to find almost like the overlapping biomarkers that would fit to everyone. However, when it comes to the, we do, again, I didn't mention this to you in here, but we are like trying to validate all of those biomarkers in different sources. So for instance, one of the studies that we are doing, we do have fresh frozen tissue samples from the patients. You can see that those biomarkers or the microRNA expressions correlate beautifully when it comes to the disease status. From the same patients, if you get the blood, and if you look at the plasma and the serum from the blood, you can see that those same microRNAs are not as specific as the actual tissues. So that means we do need to expand the study. We do need to have more patients if we are talking about the circulating microRNAs, and then to be able to perhaps like validate more whether these microRNAs are specific and sensitive enough.
Thank you so much for your answer. I'm moving on with the question from the YouTube participants. In microRNA research, clinical and methodological heterogeneities are observed that can arise at many points during the study. Can a standard protocol be created to reduce this heterogeneity?
Okay, another great question. So yes, that is one of the challenges that I was trying to mention. So the opportunities are great, but we also need to tackle the challenges. So to be able to tackle that challenge, I believe we need to have a panel of microRNAs, not only microRNA 21 or 155, but we have to have like 3 to 5 microRNAs, and we need to look whether the patients are like expressing the same trend of this panel of microRNAs, rather than like focusing on a magic bullet in a way, like in one single thing.
Thank you so much. Another from the YouTube participants. Do the prognostic association of microRNA 21 differ when analyzed within breast cancer subtypes in TCGA?
Yes, wonderful, wonderful, Seçil. Thank you so much for picking that up. When we look at the TCGA, we looked at the all available breast cancer patients' data. So it was like almost, as I said, 995,000 patient samples we looked at, but we didn't group them in terms of the PG, progesterone positive, estrogen positive, or triple negative. It does differ. So we didn't do that in that paper, but then again, reviewer asked and then we looked at it. So you're absolutely right, Seçil. I didn't show it in here, but we do see there is a higher expression level in the triple negative breast cancer than the hormone dependent patient samples.
Thank you so much. I'm moving on to the next question. In your study, microRNA 21 expression levels were positively correlated with reduced overall survival status. There was no significant result for disease-free survival ratio. Have you any opinion on what means this result?
Okay, good question. No, I don't have any opinion because I am more interested in the overall survival rather than the disease-free. Remember, all of these studies are mostly focusing on the metastasis. So we are mostly focusing on extremely aggressive cancer types. So I guess to be able to answer your question, we need to go back, perhaps the early diagnosed patients, and then look at what they are doing in the disease-free, like during after their treatment. But particularly we as a group, we haven't done that.
Thank you so much. I'm moving on to the question from the YouTube participants. When we think late diagnosis and resistance to chemotherapy in OC, how can we occur clear communication between tumor microenvironments, chemical compounds, and carrying cargo stability?
Very loaded question, Sevginas. Thank you so much. I guess we do need to look at the different parts of this, right? So with the tumor microenvironments, which we are currently doing, so we need to create the organoids to be able to tackle that problem. Because so far, almost all of the studies that I showed you, they were done either with the patient samples and or the 2D tissue or the cell culture studies. So we are already doing 3D cell culture, but to be able to answer your question, we do need organoids, not only the cells, pancreatic cancers or breast cancer cells, but we also need to have the macrophages and the fibroblasts. So we do need to create a similar model to be able to answer this question. And then the microenvironment chemical, I guess once we have the organoids, we can also treat them with the cadmium, nickel, or lead, or whatever environmental toxins that we are talking to, we can treat these organoids with them and then try to understand the mechanism better.
Thank you so much. I'm moving on to the next question. Is efficient modulation of downstream targets in the therapeutic scenario of microRNA 21 dependent on different regulators such as cell migration related genes?
Okay, another good question. I tried to show you like 3 different studies, but I didn't show you the full papers. So if you are more interested, I definitely suggest you to go and read the paper. But with the migration, cell migration, we did look at the EMT markers in the epithelial mesenchymal transition and the cell migration. We didn't look at the gene expression. We just like did the scratch assay for the breast cancer study. For the prostate and the pancreatic cancer studies, not only the scratch assay, but we also did the invasion assay. So these 2 are quite different than each other, because the scratch assay shows you whether the cells are migrating, moving or not. Another one, the invasion assay, you know that with the invasion, the cells, they do need to secrete the enzyme, the lytic enzymes, to be able to invade the matrigel. So we've also done that, but we didn't look at any particular MMPs, for instance, matrix metalloproteases.
Thank you so much. I'm moving on to the next question. Is it known which genetic changes produce traits in cells that can metastasize? If known, what are they?
With genetic changes. So it is quite difficult to point out only 1 or 2 genetic changes. As I try to summarize, there are like so many things going on. So there is like the cell signaling pathways are activated, and most of the cancers, there's a genome instability. But more importantly, there are like new fusion genes occur as well. And I told you in the prostate cancer study, although we didn't look at specifically at the TAMPAS-ERK, but in one of my previous studies, we were looking at the TAMPAS-ERK fusion, which is an event almost like 60% of the prostate cancer patients. That very much involves in the gene expressions that those genes are identified to regulate metastasis. So it is a great question, Mehmet Emin. However, it is quite difficult to point out 1 or 2 genetic changes. So there are so many of them.
Thank you so much. I'm moving on to the next question. In your study, could you identify the possible mechanisms of GATA2 regulation by microRNA 21 in breast cancer?
Great question, Elif. Thank you. GATA2 is a transcription factor, and it is quite important. We are looking at the GATA2 as well, especially it is important for the cancer stem cells. So yes, we did find, but it is, egg or chicken problem again, right? So which one comes as a first event, whether the microRNA higher expressions are triggering all of those like abnormal cell signaling pathways, or these signaling pathways are already started to get mad, and therefore the microRNA 21 sort of pushes them to even faster to go to the dark side. That is a bit difficult to assess right now, but you're absolutely right. There is an association and regulation between the transcription factors and the microRNA expression in breast cancer.
Thank you so much. I'm moving on to the next question. Is early diagnosis possible with microRNA quantification, which will be widely adopted in primary healthcare? What are its limitations?
Wonderful question. That is my aim. So we are trying to use the microRNAs, or we are trying to propose the microRNAs, not on only one, as I said earlier on, as almost like a panel to be able to use in the clinic, perhaps like as a confirmation studies or the confirmation tests, because there are like lots of great things about the microRNAs, although there are challenges, the limitations. The major limitation in my opinion, is how to collect the samples. So if we are going to use the blood samples, obviously you need to collect the samples. You do need to use it in a certain tubes because the hemolysis is a problem. You need to spin it down. You need to, if you are going to keep it in the plasma or serum, you need to keep it in the -80s or -20. So in my opinion, the biggest limitation or the challenge is the collection of the samples and also the storage of the samples. If we are going to use the tissue samples, this time by, again, I think I showed some examples of the immunocytochemistry or no, in situ hybridization. This time, as soon as during the surgery, the tumors or the tumor samples tissue samples are collected, they do need to be in the formalin. So you cannot keep it in the room temperature because this time the RNA quality is going to suffer. So the limitations are the storage.
Thank you so much. Could the EMT pathway being downregulated by inhibition of microRNA-21 lead us to say that microRNA-21 is a possible target to inhibit cell invasion?
Good point, Hilal. I think yes. The short answer is yes. In 3 different studies and 3 different cancer types, solid tumor cancer types, we did show that when there is no microRNA-21, invasion reduces. But remember, all of these studies are like in vitro. We haven't done any mouse models in terms of like checking whether the microRNA, removing the microRNAs are going to reduce the metastasis. So in 2D cell models, the answer is yes.
Thank you so much. I'm moving on to the next question. Do microRNAs play functional roles in complement-mediated tumor immune escape? How can microRNAs be exploited to increase complement-mediated cell destruction? Thank you.
Thank you, Hilal. It's quite a good question. So I don't think the microRNA's functional role is well known enough in terms of the immune response. So there are like other targets for the immune response. So in the map that we sort of like put microRNAs in the cancer hallmark, we did identify or named a couple of microRNAs depending on the available studies. But first of all, we as a group never looked at the immune escape. And 2, there are like more imminent factors for the immune escape rather than the microRNAs.
Thank you so much. I'm moving on to the next question. Many microRNAs have already been reported to be promising biomarkers for certain types of cancer, but they have a specificity issue. Is there any solution in this problem?
Yes, Elena, you're perfectly correct. There is a specificity issue, but the answer is looking at more than one microRNAs and perhaps looking at the association between 2 microRNAs, as we do it with the EMT pathway, for instance. So just come up with like 3 or 5 microRNAs as a signature of that particular tumor and see whether those 5 microRNAs are already upregulated or downregulated.
Thank you so much. Can microRNAs be used as a therapeutic target to increase the efficacy of radiotherapy and reduce radiation-induced malignancy?
Wow, another great question. So you guys are brilliant. Thank you, Sedanur. It is a very good question. So the first part is yes. Can microRNAs can be used as a therapeutic target? Yes. So there are lots of companies are working on it. The radiotherapy, I'm not sure about that. I don't know. I've never seen any study that they can use the radiotherapy or to reduce the radiation-induced malignancy with the certain treatment of the microRNAs. I'm not aware of any study, but hopefully in the future we'll see more.
Thank you. There is considerable research into the use of panels containing multiple microRNAs. However, how much do you think microRNA interactions are taken into account here?
Yes, Saida, you're also quite right. So there are lots of research going on. So some of the research now are not only looking at microRNA interactions, but like as the lots of you already mentioned, to increase the specificity and the sensitivity, we are also trying to identify link RNAs, long non-coding RNAs, and look at the link and microRNA associations, not only microRNA, but also the another layer, link RNA and microRNA association. And yes, we are looking at those MER and MER-to-MER interactions as well.
I would like to thank you for your informative answers. Lastly, I would like to ask that what advice would you give to students studying in scientific fields?
Read more, ask lots of questions, keep asking questions, and read more, and definitely be curious.
Thank you so much.
Thank you so much. Great to have all of you, and thank you so much for all those brilliant questions, and enjoy the rest of the day. Thank you.
Thank you so much. I wish you healthy days.
Thank you.